Get started. So, good morning. I'm your substitute teacher, Professor Brian Cunningham. So, Gabby asked me to fill in for him while he's away, I suppose, and happy to share with you another form of microscopy that those of you who are not in my group may not have heard about before. So, I think it's probably appropriate for a microscopy class to share with you a form of microscopy that, rather than using a piece of glass as a surface upon which you're doing your microscopy, in our case we're using a nanostructured surface, a photonic crystal, to do different types of biological imaging of cells with a technique that we call photonic crystal enhanced microscopy. So what I hope to do today is to tell you a little bit about what a photonic crystal surface is, what the properties of them are, and on how we can use those properties to enable this new form of microscopy. And I'll tell you what photonic crystal enhanced microscopy is. So I'll describe a few applications. We have many applications that we've been developing, but I just picked a couple representative examples where we're using it for doing label-free imaging of cell surface attachment. And then where we can use the photonic crystal to enhance fluorescence output to do something that we call photonic crystal enhanced fluorescence microscopy. In this class, have you already learned about TIRF, for example, total internal reflection fluorescence? No? Okay, so all right. I was going to say it's similar to TIRF, but that now it doesn't matter. So the surface that we're using in our work is called a surface photonic crystal. In optical circles, some people call the same device a guided mode resonance filter. And so what it's comprised of is a low refractive index substrate that could be made out of plastic or glass. We'll see both types of examples here. Actually, I think we'll have glass or plastic ones in a lecture today that has a periodic surface embedded into it. This is a low refractive index material on top of which we put a high refractive index material such as titanium oxide. In our work we use titanium oxide even though other high refractive index materials will also work, such as silicon nitride, for example. And so if in most of our work we make what we call a one-dimensional photonic crystal, where this is the cross-section view, but if you looked at it from the top you would see a series of lines and spaces between the lines where we have this periodic structure that repeats over and over again. And so with this, a photonic crystal is different from an ordinary diffraction grating from the standpoint that we design it to act as what we call an optical resonator, where this structure is designed to interact very strongly with a very specific band of wavelengths. And so by choosing the period of the photonic crystal, by choosing the refractive index of the plastic material and of the titanium oxide material, by choosing the thickness of the titanium oxide, we can engineer the structure to have a nearly perfect reflection at only a narrow band of wavelengths. And so here I am just showing a reflection efficiency as a function of wavelength, where at this resonant wavelength, the structure acts as a 100% efficient reflector, but at the other wavelengths, the light mostly transmits straight through. And so by choosing the period of the structure, we can make this resonance happen at almost any wavelength that we want. And in our work, we've built them to anywhere from ultraviolet wavelengths all the way out to mid-wavelength infrared. In the work that I'll show you today, we've chosen to make our resonators in the visible part of the spectrum, but here I just chose to show you one in the near infrared around 865 nanometers. So at that resonance wavelength, something unusual is happening, right? And so if we were to look inside the photonic crystal, and if we could visualize the electromagnetic field strength as a function of time at the resonant wavelength, this is what we would see. And so the light at the resonant wavelength is captured by the photonic crystal, which tries to turn the light so it can travel sideways. So the light looks at this region as a net high refractive index region that would ordinarily be a waveguide. So in a waveguide, you would have a high refractive index substance surrounded by low refractive index substance, and the light will be guided by the high refractive index material. But here we don't actually have a waveguide. But instead, light that tries to travel sideways is going to encounter some interference at every half wavelength and get re-reflected back in on itself. So light that tries to travel sideways will get back reflected here, back reflected there, back reflected there. So light doesn't actually have the ability to propagate very far in the structure like it would in an ordinary waveguide. So what happens instead is all these back reflections cause a electromagnetic standing wave where we see the light going from, we see the electric field going from positive to negative and positive to negative over and over again, but confined mostly at the surface. And so what you see in this electric field standing wave is that some of the energy is actually in the medium, in this case water, that's next to the structure. And so what we've done now is to devise a way to take light from an LED or a laser, confine it to a surface, and to amplify its value, to amplify its electric field intensity. But that electric field intensity is confined to a couple hundred nanometers from the photonic crystal surface. So if we were to take an average of this over many periods, we could think of this as what's called an evanescent field, where the electric field is near the surface, but is declining in intensity as you move upwards and away. And so in this case, if we illuminate the structure with an electric field magnitude of 1 volt per meter, what we would see is electric field that's constrained to the surface with electric field value that can be much, much higher, and even hundreds of times higher. And so this evanescent electric field is what we hope to use for interacting with biological stuff on a surface. So in our work, we use these photonic crystal surfaces routinely for doing biosensing work. And so it's a structure that has nanometer scale features, and so it's important to have a way to make them inexpensively, so we don't have to go to the e-beam lithography system every time we want to make a sensor. Usually we use them once and then throw them away. So we developed an approach called replica molding, by which we take a glass substrate and we put a small amount of liquid ultraviolet curable epoxy on the surface, and then we take a silicon wafer that we've etched an inverse of the pattern that we want to create in our final structure, and we mush that into the UV curable epoxy. It fills in all the grooves on the silicon wafer, and then we can illuminate with ultraviolet light from the back to cure this liquid epoxy into a solid. And then after that, we can remove the silicon wafer away. And so we can do this kind of process using big silicon wafers, even up to 8-inch diameter silicon wafers. And we can make these structures very uniformly, very reproducibly. Then using your favorite method, could be evaporation or sputtering, add titanium oxide to the top. And then, so the typical dimensions for our visible wavelength photonic crystals is a period of around 400 nanometers, height of the grating maybe 120 nanometers, and thickness of titanium oxide 60 nanometers. Then if we want to, we can add other things to the top of this titanium oxide. So for compatibility with cells, for example, we may top this off with a few nanometers of silicon oxide, so the upper surface appears to cells like glass, like it would normally encounter if it was on a petri dish or a glass microscope slide. Yeah, so for the work I'll show you today, we're making photonic crystals that are around 9 millimeters square. Here's one just on a 1 by 3 inch microscope slide. And so how do we use a structure like this for label-free detection? And so what I mean by label-free detection is that we're interested in being able to detect cells or biomolecules. In this case, I'll be mostly talking about cells, through their intrinsic properties, not using dyes or stains. So if we want to detect them by their intrinsic properties, let's think about how we might do that. So if we have our photonic crystal reflection, and so here I've drawn it so that we have a high reflection and a narrow band of wavelengths around 620 nanometers. If we take an object and put it on the photonic crystal surface, but let's imagine that this is a dielectric object. What we've done essentially by taking this dielectric object and moving it from here to there is that this thing has replaced some of the water. And so instead of water being here, now we have this object that has perhaps higher refractive index than the water it replaced. So when we put something here that has a higher refractive index, we'll get a shift in the resonant wavelength from a lower value to a higher value. Basically what we've done now is to modify the coupling condition for light to the photonic crystal. So now a higher wavelength now satisfies that resonance condition. And so we call this a change in the peak wavelength value. Now, if this dielectric object happens to be an absorber, if it absorbs the resonant wavelength, or if it can scatter out the resonant wavelength, in addition to seeing this wavelength shift, we'll also see a reduction in the resonant intensity. What I mean by that is that the reflection efficiency will drop from 100% to some value less than 100%. So we call this a change in the peak intensity value, or the PIV. So going forward in the talk, you'll see peak wavelength value shift. That's the change in dielectric constant when we bring something to the surface. Or we may see a peak intensity value shift, or a PIV shift, if our attached object is an absorber or a scatterer of light at the resonant wavelength. And so what we'll focus on today is how we use this photonic crystal for cell microscopy. And so there's a few different reasons that we might be interested in doing this. In the world of cell imaging, you may have already seen a lot of this in class. You need to have something that gives you contrast because cells don't have a lot of native contrast. So one of the things that Professor Popescu's group specializes in is using phase contrast microscopy, where the cells have refractive index that slow down light a little bit, and if you engineer an instrument that can allow you to see cell bodies by their refractive index contrast, that is very helpful. In our case, we're doing something very similar, and for similar reasons to why Professor Popescu's group has phase contrast microscopy. One is that fluorescent dyes have a very limited lifetime. If you've done fluorescent microscopy, you add your fluorescent dyes to some cells, you put them under the fluorescent microscope, and you better act fast because you've got about 30 seconds before the fluorescent dyes photo bleach and they go from being observable to being dark again. Unfortunately, cells do very interesting things after 30 seconds that you start looking at them. And so cell processes like apoptosis, stem cell differentiation, a cancer cell response to drugs, some of these things take hours or even days to manifest themselves. And so people are interested in processes by which you don't have this time limitation posed by fluorescent markers. Other thing is that you can stain cells with chemical dyes that would make them visible under an ordinary bright field microscope. But unfortunately for that, these kind of stains are toxic to cells, so you can look at dead cells. And so for many things, people are very interested in looking at live cells and live cell behavior that can take a very long time. And so here, I just kind of show as an example, we are interested in looking at what happens at the cell membrane and the cell membrane's interface to extracellular matrix. And so I think that this is what differentiates what my group does from what Professor Popescu's group does. So whereas his group can look at the entire cell body, we're entirely focused on what happens at the cell membrane interface. For example, cells in the body, most of them, except for the ones that are floating around in your bloodstream, for example, are what are called attached cells. They are attached to a basement membrane as part of tissues, and so the ways that cells interact with cell membranes is very fundamental to biology and understanding many of the processes that cells undergo during all these biological processes. I'll mention here like one of the things that we can do with a photonic crystal that's kind of interesting. If we look at our photonic crystal and we take a fixed wavelength, so I showed you first that we can take like all wavelengths and under normal incidence and get this resonance reflection. We can also, if you want to, choose a fixed wavelength, and then this coupling condition will be satisfied only for specific angles. So in this way, we can take a photonic crystal that has high reflection intensity at specific angles. We can choose to, if we want to illuminate the photonic crystal either on resonance or off resonance just by choosing the angle of incidence. So this is a concept that I'll use later. So if we want to use our photonic crystal for fluorescence, so I just said fluorescence is bad, but it's not like a religious belief. People still use fluorescence in plenty of cell-related work. And so we can use our photonic crystal for fluorescence by illuminating on resonance, where we have this big electric field on the surface. So here, just to show you the difference between on resonance and off resonance illumination, this plot shows a electric field magnitude as a function of position in one period of the photonic crystal, where here's our water on the surface, our titanium oxide and silicon oxide, where you check out the magnitude of the electric field. So on resonance, the electric field can be in the hundreds of volts per meter when we illuminate with 1 volt per meter, whereas off resonance, the electric field magnitude is close to 1 volt per meter that we illuminated with. So we can switch between on and off to expose any fluorescent material here to either high field or low field, and either turn its brightness on or off. Another interesting feature is that the electric field magnitude drops off as you move away from the photonic crystal in a very predictable fashion. It has an exponential fall off like this. And so I'll show you how we can use this on-off feature and our knowledge of the electric field magnitude drop-off to help us estimate the position of fluorescence emitters above the surface. And so in ordinary fluorescence microscopy, you just look at fluorescence intensity. Our method of being able to tune on and off and to use this known electric field profile allows us to actually estimate the position of fluorescent dyes relative to the photonic crystal surface. Okay. So let's go back to label-free detection again, where I mentioned we can take a nano object or a big object, put it on the surface, and we have these changes in peak wavelength value or peak intensity value that we can measure. So how do we integrate this sensor with microscopy? So here's in our laboratory the photonic crystal microscope. So it looks kind of like a regular microscope from the standpoint that we have an ordinary microscope body, but instead we have a spectrometer on the side over here. I'll show you how we use that. And we have our illumination, you can't really see it, it's kind of through the back of the system. But schematically it looks kind of simple. So we take a light emitting diode, we filter out one polarization because we only want to illuminate our photonic crystal with a light that's polarized along the grating direction. That's the direction that gives us that resonance. We reflect that light, we put that light through a cylindrical lens, so the light is focused down to a line, and we put that line through a microscope objective that illuminates the photonic crystal from below. The photonic crystal has that resonant reflection. The reflection comes back, goes through that same microscope objective, but we put it through a dichroic mirror into a imaging spectrometer that allows us to measure the peak wavelength value and the peak intensity value on every position on a photonic crystal surface. So how do we do that? So let's imagine this thing is our photonic crystal surface, have the grading lines going that way, and that these little smiley faces represent some cells that are on the photonic crystal surface. So I mentioned that we take our light emitting diode and we focus it down to a line. So here's our line of illumination on one position of the photonic crystal surface. So if we take one pixel out of that line, that pixel is represented by a reflection spectrum like I've shown here. By reflecting the light into an imaging spectrometer in one snapshot, actually we gather a spectrum for every position all the way across this line. In our system, we have a spectrum for every half micron increment in space going in this direction. And so in one snapshot from our CCD camera, we measure 512 spectra, each spectra representing one place along the line. Make sense so far? Okay, so now imagine we take this line and we actually what we do is move the photonic crystal underneath the line in small increments, so that step by step we build a big data cube where each position in the y direction has 512 spectra associated with it. And so we have the ability now to go into all this data and pick out the peak wavelength value and the peak intensity value for all 512 by 512 spectra. And so what I show here is it looks like an image, but it's not like really an image like you would ordinarily get from a camera. Each pixel in this image is represented by one of these spectra, where each spectra has a peak associated with it, within which we can analyze the peak and find, for example, the wavelength of maximum reflection. And so this image here for some pancreatic cancer cells, ordinarily this would what they would look like in a bright field microscope image. In a photonic crystal microscope image, this image represents the contrast bar represents wavelengths of the maximum reflection. So for example, in a place where there is no cell, I call it the background, would have a lower wavelength. A place where there is a cell will have a higher reflected wavelength. And so if we zoom into this example image, you can see some interesting things. And so, for example, we can very easily observe the cell attachment footprint, as we call it, and how different cells have even supposedly identical cells here growing on a photonic crystal surface have very different footprints. That even within an individual cell can have regions of low attachment strength, regions of higher attachment strength, and interesting shapes in terms of how the cell is attaching to the surface and starting to spread out. Questions about that? Okay, so what we found is that cells have very interesting and distinct cell attachment behavior that we can observe with the system. And so here I'm just showing a few different types of cells and their attachment footprint as measured by the photonic crystal microscope, with the color bar representing the wavelength of maximum resonance. And so here's an example of one of the things we can do looking at stem cells differentiating over an extended time period, in this case over 10 days. And so our instrument is integrated with a cell incubator, so we can keep the cells in the instrument and alive for many hours or many days. And we can take snapshots of their cell attachment for many different times. And so, for example, we see that cells initially attach to a blank photonic crystal by small attachment points. Those attachment points spread over time. Cells do some kind of wild stuff. And so, for example, here's a cell that has a strong attachment in the center, followed by like a corona where there's no attachment, followed by heavier attachment around the outside. Here you can see that the same cell 10 days later has like a center area, a little gap, and then more cell attachment around the outside. So interesting things are going on in there in terms of how the cells attach, and how within an individual cell, how the cell attachment varies from place to place. So since we can take one image, we can take many images. And so, for example, here at 10-minute increments, here's stem cells attaching versus blank, an initial attachment point. The initial attachment point spreads. The interesting thing you can see here is that there's a cell here. You can see that the cell is there in the bright field microscope image, but it hasn't attached. And so what we see is that as the cell forms a close association with the photonic crystal surface, it actually starts to register these changes in wavelength. You can also have cells that kind of hover around and sit near the surface but don't actually attach, that they're like physically present but they haven't attached. And so we can actually even see how this one attached spreads, gets joined by its brothers and sisters later. We can do things like make dynamic movies of the cell attachment, like outline, how that changes with time. And we can even dive into these images one pixel at a time to see how even one pixel here, if you look at the red one is, so here like the black line is for the cell that's shifted, for where a cell is attached. The green is for like the background area. The cell is going to spread over the red pixel, and you'll kind of see that the red pixel shifts from being over on the left to being over on the right once the cell kind of goes over that point. So we believe pretty strongly. I think we've established that these peak wavelength value shifts are definitely associated with the cell membrane and the cell attachment over these places on the photonic crystal surface. And so with that kind of thing, we can make photonic crystal attachment movies where this shows one of those cells attaching and then spreading as a function of time. And we can even make like movies of the cell boundary and how it evolves with time. Here's kind of an interesting experiment where we have some stem cells attaching to a photonic crystal. Here we were taking images every 2 minutes, and so you can kind of see them attaching. You can see the peak wavelength value being like relatively higher in some places rather than others. What we're going to do is after these cells attach, we're going to kind of skip ahead in time. So now we have lots of cells attached. We're going to expose these cells to a drug called trypsin that kind of shocks the cells and they jump off the surface. So you kind of saw them like all go off the surface, then they kind of start to reattach, find their way back to the surface. And so we can clearly see that these images that we're gathering represent actual cell attachment and the cell attachment footprint. I think we have one more trypsin exposure. They'll kind of do their jumping thing again in just a minute. Yeah, there they go again. So we've done like other kind of little cell tricks. This is one where we did a chemotaxis experiment where we put up here out of the field of view an agarose bead that has a chemical attractant to this attached cell. If I can figure out how to, no. Or maybe I won't do that. I need to find my pointer. Rats. Oh, there it is. Okay, so this cell is going to gradually kind of move from south to north as it senses something in its environment and is going to actually like crawl itself in one direction. One thing that we found is that we really use this as an imaging modality using the wavelength shifts initially. And so we, our hypothesis was basically that the cell membrane and its components, when they attach to the photonic crystal, those cell membrane components are engaging with that evanescent field and displacing water that would ordinarily be there with high refractive index stuff. That's like the lipids in the cell membrane, the proteins that are behind the cell membrane, and that's what we could observe with photonic crystal microscope. Yes?
Why is there a wavelength shift? Where is the energy going? Is it going into heating up the cell, or because I assume you're getting lower energy photons coming out, right?
So what we're observing is just a change in the coupling condition for matching the resonance between the incoming light and the photonic crystal.
The light coming out doesn't change in wavelength.
That's right. Yeah. So you're not really changing the light itself. We're illuminating with a broad band of wavelengths from an LED, and only in the photonic crystal only reflects back a narrow set of them. So that's actually one of the good things about this system is that we can illuminate with low intensity light from a broadband source. We're not really imparting much energy to the cells themselves. So like people get concerned about like phototoxicity and using lasers to illuminate cells, where here we're using very low intensity light. So yeah, excellent question though. Any other, does it make sense? Okay. In our work, we're using the wavelength shift primarily, but we were gathering all these spectra anyway, and so we kind of asked ourselves, what would we see if we also looked at the peak intensity value? So I'd like to act like we were a genius and we thought there was all this information in the peak intensity value things that we were measuring, but in fact we kind of discovered it by accident later. And so here we're just looking at some, I think these are dental stem cells attaching with the peak wavelength value mode, first of the blank surface, and then kind of you see them attaching and spreading over time. We can look at those exact same cells, but rather than looking at the reflected wavelength, we can measure the reflected intensity, and what we see is something very different. And so if we zoom into some of those cells, what we see is that the places where there's the most wavelength shift are not the same as the places where there's the most intensity shift. And so here, these are like arbitrary units, but red represents places where there's more reduction in the reflected intensity. And so you can see that these are kind of localized in some cases around the perimeter of the cell, but not always. So here's one that has a big shift in the center of the cell, but then it changes so that it's around the perimeter. So we started thinking about, well, what's going on? Well, what are we seeing that's different in a peak intensity value shift? Here again, just zooming into one cell, and here I wanted to show you this so you get an idea of the resolution, even within an individual cell, that we can study the cell attachment footprint. So I think these images have pixels that are 0.5 microns on a side. So so we can do the same thing with movies. We can take peak wavelength value movies. And if I can move ahead, I think we have to watch the whole movie. Sorry about that. And then, or we can make a peak intensity value movie of the exact same cells. And so our thought here is that cells, as it turns out, are not like big water balloons that have uniform characteristics all around their outside cell membrane. When cells attach to a surface, the way they do it is by something called focal adhesion points. And so these focal adhesions are represented by places on the cell membrane where proteins gather up together in aggregates, not only on the cell membrane themselves, but they're backed up behind the cell membrane by things like actin filaments and other aggregated proteins that make like a link between the inside of the cell and the outside of the cell. And in fact, these focal adhesion points are how cells communicate with their environment and transduce information from the outside of the cell to the inside of the cell. And so our hypothesis, and one of the things our research group is working on actively now, is to show that these cluster areas represented by reduction in the peak intensity are due to aggregates of protein that are scattering out light more efficiently in those places compared to other places. And that in the places where we see a peak intensity value change is a place where there's a focal adhesion. One of the things I get asked almost any place I give a talk on photonic crystal microscopy is what's the effect of that grading structure, the photonic crystal nanostructure, on the cells. And so I got so tired of answering that same question that our group actually developed a way to make the photonic crystal surfaces planar so that we wouldn't have the following problem. So one thing that we would observe very consistently is that the photonic crystal grading direction, if it goes in a particular direction, we don't actually see the grading. The grading is too small compared to the cells to actually show up in our images. But the cells know it's there, and the cells have a way of sometimes sensing that environment and growing their attachment along the grading direction. And so we developed a way to fill in the grading grooves with a polymer called SU8. We developed a horizontal dipping method that would allow us to place just a very small thickness of the SU8 within the grading grooves, and then we etch it back slightly so it just barely fills in under the surface. But when we do that, we can, here just showing in an atomic force microscope image, have it go from like grading mode to planar mode. Here just showing that our grading structure would ordinarily be about 100 nanometers. After we planarize it, it's down to less than a nanometer in thickness variation. And so if I can manage it, the cells on the left are growing on a regular photonic crystal, and eventually you'll kind of see them form attachments with the cell grading direction going up and down. Whereas on the planarized one, we see a behavior that's more evenly balanced. They don't seem to sense the grading direction anymore, and we will see them spread out in left and right directions and up and down directions about equally. Okay, so I think just in the last few minutes, I wanted to share with you one more thing about fluorescence and fluorescence enhancement using cells in this photonic crystal microscope. So I alluded to it a little bit earlier. Label-free detection is fantastic. Fluorescence is still heavily used in cell imaging, and we have this ability with the photonic crystal to amplify the electric field and to boost fluorescence output. So we thought, why not see what we could do with fluorescence enhancement using photonic crystals and using this same microscope. And so we have this ability to tune between the on-resonance and off-resonance condition by choosing the incident angle if we used a fixed wavelength. And so here they're just choosing a monochromatic light source, such as a laser, to illuminate. Now there's specific angles that wavelength will couple into the photonic crystal. So it kind of repeats what I said before. So what we did was to incorporate a second illumination path into our instrument. So this is the illumination path that I told you about before for doing label-free imaging. What we added was this, where instead of an LED, we have a laser. The laser goes through an optical fiber that expands, gets collimated by a collimating lens, and then we put that, we have the ability to choose between either label-free imaging or fluorescence imaging using this mirror. We can put the light through the same cylindrical lens, focus it down to a line, and then illuminate our cells from below on the photonic crystal. Now one thing that very important is actually here with these sneaky arrows. So we have the ability by this cylindrical lens, its focal point is on the back focal plane of that objective. So I'm not sure if you learned this in microscopy class, but if we change the position of this focus point to be off the focal plane of the objective, we can manipulate the angle of incidence of light that emerges from this objective. And so what we have in our system is a way to move this cylindrical lens. The cylindrical lens sits on a computer-controlled motion stage, so we can translate it. And by translating it, we translate the position of the focus point. And by translating the position of the focus point, we can manipulate the angle of incidence on the photonic crystal. Does that kind of make sense?
There are people doing that for light sheet now.
Yes, yeah. So same basic thing. I think we may have got the idea by talking with Professor Popescu about this. So with the system now, we have the ability to quickly switch between on-resonance illumination and off-resonance illumination using that laser source. And so here is fluorescence images. They're also taken by line scanning. But rather than gather spectrum, we just gather fluorescence intensity as a function of position. We don't have to measure peak wavelength values or peak intensity values. We just measure fluorescence intensity for the light going this way. So we don't use our spectrometer as a spectrometer anymore. We just measure the fluorescence intensity. So here's some fluorescence images of mouse fibroblast cells attaching to a fibronectin-coated photonic crystal, where we used a stain called a cell mask that's supposed to only preferentially stain cell membranes. And so if we take a off-resonance image of these cells, we can see the places where there's more fluorescence intensity. If we then change the illumination angle, we can get an on-resonance image of the same exact cells. So not much really changed here, except you notice that they went from lower intensity to higher intensity. That's our photonic crystal enhancement happening. So we can do something with these two images, the on-resonance and off-resonance image. We can take one and divide them by the other on a pixel-by-pixel basis to make what we call an enhancement factor image. So an enhancement factor image is I guess what it is an image of what the enhancement factor is for each of these places in the image. So what do you see in an enhancement factor image that's different than what you see in a regular fluorescence image? Well, a fluorescence image tells you something about the amount of fluorophore that's within each pixel in the image. The more fluorophore stuff that you have gathered in one place, means more intensity, right? Now, if we take an enhancement factor image, what we've done now is to normalize out the amount of fluorophore. And what we've done instead is to weight the intensity of this image by how close the fluorophore is to the photonic crystal surface. Since we know the mathematical equation that describes the drop-off in enhancement factor as you move away from the photonic crystal, we can take this enhancement factor image and make an estimate of the position of the fluorophores from the surface in units of nanometers. So with somewhere on the order of nanometer resolution, we can say with an enhancement factor image how close a fluorescence emitter is from the surface. So we did this with a membrane-based stain. We did the same thing with a nucleus stain. So here's a stain called Nuc Red. It's supposed to stick to nuclear components, only stain the nucleus. And so we were wondering what we would see in an image like this, because obviously the nucleus is not in the cell membrane, it's behind the cell membrane. And so, but still we could see like an off-resonance image and an on-resonance image, so image of the same cells, just higher intensity here, make an enhancement factor image, and then actually see within the cell how close the nucleus is to the cell membrane underneath it. So to conclude, we're using these nanostructured photonic crystal surfaces to do some unique things that aren't available in other forms of microscopy. And so one of them is label-free imaging, where we have this ability to take images of the peak wavelength value or the peak intensity value. It's a label-free approach that is focused specifically on what happens at the interface between the cell membrane and the surface it's attached to. And this is a biological situation that's very common for many types of cells that people are interested in. People are also interested in knowing where things are within cells and their distance from the surface. And so if you can label something with a fluorescent dye, you can use this enhancement factor image modality to help locate things, at least in the Z direction, with very nice resolution. Of course, the students in my group, I need to thank them profusely, and I'm glad there's 4 of them here today. Now, none of you all are doing fluorescence microscopy kind of work, but you never know. So obviously the students in the group did all the great work that I showed you here today. Of course, we have funding from National Science Foundation and NIH to help us along. So happy to answer questions. Really appreciate your attention today. Thanks for clapping. You don't have to clap in class. But.
Is there any specific reason why you're using line scanning? I think it has the time resolution, but it compromises the spectral resolution because the CCD is not actually like spectrometer.
So the CCD, maybe I didn't explain clearly enough, but the CCD actually is functioning as a spectrometer. So. Yes. So it's a I guess what you would call a imaging spectrometer. I can find this. Sorry. So yeah, it's an important point. So I'll try to explain when we get to the instrument here. Yeah. So what I didn't explain very well is that the spectrometer has a slit that the line of light enters. Behind the slit is a diffraction grating. And so the diffraction grating spreads out the light along its spectral components. So in one dimension of the CCD image is spatial. The spatial dimension of the CCD image represents like a place. The other dimension of the CCD image represents wavelength. Does that make sense? So it's because we have a diffraction grating is taking the line of light and dispersing it, we're able to measure the resonance reflection as a function of for each of these pixels. Does that make sense?
So you combine that spectral range to like 600 to 640.
Yes. Yeah, that's right. I mean, our spectrometer actually goes further than that. We only care about a narrow part of it. Yeah, so we throw away most of the data from our spectrometer and only focus on this narrow part. Make sense? Okay.
It looks like two things will contribute to the shift of the wavelength and the change in the intensity. That is the refractive index structure of your sample, and also how well the attachment is.
That's right, yes.
Like the distance between them.
Yep.
How do you separate these two effects, or do you look at them both?
Yeah, I think we look at both at the same time. So it's hard to separate one from the other, if it's possible at all. So as a cell attaches to the photonic crystal, like you say, it attaches, but then I mean, a cell has all this stuff behind the cell membrane. The cell membrane itself is maybe only like 5 to 7 nanometers deep, but our evanescent field is maybe 200 nanometers deep. So what we're really looking at is the cell membrane and whatever's behind the cell membrane. We kind of estimate more or less that like all the protein components inside a cell have like around the same refractive index, and the research kind of supports that. That if you kind of think of the cell as like a bag full of proteins and lipids and stuff, that most of that biological material has very similar refractive index to each other. But as it gets closer to the surface, its effect on the reflected wavelength is more. So we are kind of measuring a combination of how close it is and how much stuff there is at the same time. And I think that is one of the things that's interesting about the peak intensity value modality is that when there's protein aggregates that scatter more than their surrounding area, that perhaps we're able to see that. But that doesn't show up as a different refractive index. It shows up as scattering. So we're like, why is that? That's one of the things that we're like currently working on.
Have you tried like higher index materials to get better Q factors, or like for the titanium oxide, or is that like the highest that you can?
Yeah, the having better quality factor will give more enhancement. It doesn't necessarily make the amount of shifting more. So we can manipulate the quality factor a little bit by choosing the refractive index of the material, or the thickness of the grating or depth of the grating, things like that. We kind of have a trade-off. We want the resonance to be relatively easy to measure at the same time. So if it becomes really narrow, you may only get a couple pixels worth of reflection spectra out of your spectrometer, but we'd really like to be able to measure like the whole curve. Yeah, so we kind of have a trade-off between like resolution and ease of coupling light into it.
Can you talk a little bit more about the process? Is this like the epi work stuff, epitaxy, or do you use E-beam? What do you use in the lab?
Oh, to make what part?
Yeah, to make the crystal. I think you talked a little bit about the epitaxy, or is that the process?
Oh, well, we use, we make a master mold out of silicon, where we'll have that made at a silicon foundry by deep UV lithography. I'm usually using a stepper to make like a big grading master. It's also possible to go into our clean room and have a master made by E-beam lithography. There's even companies you can go to buy gratings with different dimensions. So we use the grading as a mold that we emboss into a UV curable polymer using that replica molding method that I mentioned.
Okay.
Does that make sense?
Yeah.
Great. Other questions? Okay, well thanks so much. Hope you have a good rest of your week.